about the different types of cell culture media. So as it is, to give a small introduction about cell culture media. So basically, when you want to culture cells, you have to have basic requirements like the media, then the sterile conditions of maintaining the CO2 incubator, and also the equipments. So like how you sterilize your all your glasswares for before culturing culturing the microorganisms in the lab. Similar way, we have to sterilize all the equipments that we are using for our culture purpose. Okay, because cell culture is a very tedious experiment. I mean, it looks very simple, but then once a small contamination occurs, then the entire thing is collapsed. Okay, so we have to again re-sterilize the entire incubator, and in some conditions we might have to sterilize the entire room also. So for such things, the importance is about how you maintain your lab, how you maintain the requirements of a cell culture. So if you see, cell culture is now you can see that it's a prevalent technique, and many people are getting well versed in this. And even for finding jobs in many of these companies, like biotech companies, which are having wet lab works, they prefer people who have a hands-on experience on cell culture. So from my side, a suggestion for you guys is, if you get a chance to have a small internship on cell culture technique, it will be of good to you guys. You can include it in your CV, and you can claim that you have hands-on experience on it. Okay, likewise, you can also build the knowledge about cell culture. Okay, so basically you're going to culture the cells. Okay, so that's what is written over here. Used for removal of cells from tissues or organs from an animal or plant, and their subsequent placement in an artificial environment. So that is your cell culture. So basically you're trying to isolate cells or remove cells from different tissues or organs of animals, or even plant cells can be cultured. So all these things are maintained in a laboratory, an artificial environment, by providing it with the required conditions for it to survive, and also for it to proliferate in a effective way. So if you see the cells, for growing, and it requires optimal temperature conditions, and also it requires the required pH, the humidity in the incubator. So the normal incubators that we culture microbial cells are 37-degree incubator, whereas here again the 37-degree is required. Along with that, you need to maintain the 5%, 5 to 10% CO2. So that is very important. That is the difference between your normal incubator and a CO2 incubator. Here we have external supply of carbon dioxide, which is required for your cells to grow efficiently. Okay, and along with all these parameters, the other important thing is your medium. Okay, so if you see, that is for the in vitro growth, you have to provide this medium, which is going to be enriched with different types of nutrients. Okay, for example, last week we did see about the different types of supplements that are added in the media, and also different types of nutrients that has to be enhanced with the cell culture medium. So the growth medium is very important for your cells to proliferate effectively. Okay, so growth medium or culture medium is that liquid which we saw even the video which I showed last week. We did see how they just added the powder to the sterilized water, and they mixed it to get your pink colored solution. So that medium, that is a cell culture media. So mostly it is of that red color because of the phenol red which is present in it. That's a pH indicator. Okay, so these cell culture medias mainly provide the required energy, okay, for all the cells to grow and to enter into its cell cycle. So this energy is provided not only by these supplements that we added. The media itself will have also be enriched with different types of vitamins, all the growth factors, all the amino acids that's required, and also it will have some what's the antibiotics also can be added to prevent contamination. Okay, so that's a point which is mentioned. A typical culture medium is composed of amino acids, vitamins, inorganic salts, glucose, and serum as a source of growth factors, hormones, and or other attachment factors. Attachment factors as in for the cells which attach and grow to the plastic surface of the culture flask. It requires factors which help it to bind effectively and then start its growth. Okay, so in addition to all the nutrients, that is what we have to also maintain the physicochemical properties. pH, osmolality, oxygen supply, carbon dioxide supply is also required. Okay, so please make a note of all these important things that you have to maintain for a cell culture media. So this is as it is the types of cell culture media. So animal cells can be grown both in artificial as well as natural medium. So if you see which are the natural media, those are the ones which I've listed here. Okay, there is biological fluids. So initially when people started growing cells externally, that is in the lab or in a laboratory condition, when people started the growth of cells, they did not have all these ready-made media which is available now. So they all started with using biological fluids. So fluids such as the scientists would have used the plasma of from a chicken blood or a serum from a fetal bovine. So from that they would have extracted the serum from the blood and they would have used it for culturing the cells. Okay, so these only thing they saw that all these biological fluids of course enhanced the growth of the cells, but whereas there was some ups and downs in the growth, the either the characteristics were different or they might not survive for a longer time in such medias. Okay, so even to now, you know that serum is an important factor or important component of most of the media. So I told you that serum itself is enriched with different types of nutrients and different types of growth factors. So in case it's a serum enriched media, then the addition of the supplements will be very less when compared to the serum free media, which requires more types of supplements and more types of nutrients which has to be added to the cell culture media. So you can see that these are the biological fluids which were used as media initially, that is plasma, serum, lymph, human placental cord serum. So this human placental cord serum is a very rich source of different kinds of nutrients. Okay, amniotic fluid is also a very enriched source. So these things you can understand that it helps in the development of a baby. Okay, so for the growth of the fetus, these amniotic fluid and placental cord serum provides a lot of nutrients. The supply of nutrients is through this. So in that same way, you can understand that, you can correlate and say that these fluids are enriched with all the types of nutrients that's required for a cell to grow as well. So that's why they were used as the natural media. Okay, then tissue extracts were also used as natural media, that is extract from livers, from spleen, from tumors, leukocytes, and bone marrow. So you know how rich bone marrow can be. Okay, so bone marrow was also used as a natural media. Then extract of the bovine embryo. So bovine embryo itself on the chick embryo itself were used as a natural media, as in all the required sources for all these nutrients were obtained from these embryo extracts. Okay, then clots were also used as natural media, that is clots and coagulants or plasma clots. So they were also used as natural media. So why? Because even the plasma clots are enriched with different types of nutrients and growth factors are present in them. Okay, that's why they are important. Then the artificial media, if you see, are mainly the ones which are available now. So artificial media, of course, balanced salt solution, the PBS, DPBS, all these salt solutions are the prior basis for your cells to grow. Why? Because here before culturing the cells in the particular media, the tissues or organs or cells are actually suspended in the salt solutions, which will enhance the cells to adapt to the environment, adapt to the laboratory environment and initiate the growth. So that's why balanced salt solutions are used as artificial media. Then basal media is MEM and DMEM. So they are actually minimal enriched media. Okay, so DMEM is Dulbecose minimal enriched medium, and then we have complex. These are the basic ones. So that is for simple culture or cells from a very basic source. Such cells can be such primary cells can be cultured using DMEM and MEM. Okay. MEM is again minimal essential media. Then we have complex medias. Complex medias are the RPMI. RPMI full form, anybody can tell? Anybody? RPMI is full form? Nobody knows, huh? Or everyone knows? Anybody, people? RPMI is full form. RPMI 1640. I did mention in the first class. Nobody knows. Okay, RMI is Roswell Park Memorial Institute. So it is, this medium is named after the institute from which it was developed. Okay, then we have IMDM. So all these mediums, they actually, these two are the complex media because they enhance in the growth of a wide variety of cells. That is, cells from different sources can be cultured in these two types of media. Okay, so these are just to list a few. Then again, to see in detail about what is a natural medium used for and artificial medium used for. Okay, natural media is consists solely of naturally occurring biological fluids, and they are very useful and convenient for wide range of animal cell culture. The major disadvantage of natural media is its poor reproducibility. Okay, so that's the main drawback with the natural media. So as I mentioned, the first two lines is talking about the importance of why natural media can be used for culturing cells. Okay, then the disadvantages, it is that is, it is of poor reproducibility, as in we cannot have too much. Okay, there's no excess production of this media. Okay, that's why. And the exact knowledge of composition is not known. Okay, so these natural medias can vary in their composition. So we don't know how much nutrient is there, how much vitamin is there, how much minerals are there. So that's not known to us. So that's why there is not much information about its composition. Okay, artificial media, yes, we know its composition, we know how to prepare it by ourselves. Okay, such things can be done. So artificial media or the synthetic media are prepared by adding nutrients. Okay, both organic and inorganic nutrients are used, and other things which are added are vitamins, salts. It's enriched with oxygen and carbon dioxide, and then serum proteins, carbohydrates, cofactors. All these things are added to the artificial or synthetic media. Then differential artificial media have been devised to serve the following purposes. Which are the following purposes are: first is immediate survival. So that's what I mentioned. Immediate survival, a balanced salt solution is used with a specific pH and osmotic pressure. So basically a balanced salt solution, so like once if you're dissecting a mice, okay, a mice or a rat, for example, you've gone in for the dissection room, you're dissecting your mice and you're trying to retrieve a small tissue of any particular organ for that matter. Okay, you want to isolate the cells and you want to culture it. So when you're dissecting, it's not like you can take the tissue from any part of the mice and you can immediately go in for culturing. Okay, that is not possible because you have to do the procedure. You have to complete the dissection process. You have to discard the mice. You can't leave the mice and then run into the culture lab and do your culturing. Okay, so in that way, so initially, once you have dissected the mice and once you have taken an explant will be a small portion of the tissue from which region you want to culture the cells. Once you've done it, the first thing you will do it is you'll put it in a balanced salt solution for it to get adapted to the condition as well, and also for it to survive. The cells should not die. If you leave it, if you just remove a small piece of tissue and just leave it on any glass plate and leave it till you finish your other works, you cannot get good amount of cells from it. Okay, for the cells to survive, we have to keep it in this balanced salt solution for it to get adapted to the nature, and also for the cells to survive till you go and do the culturing process. So you know that the culturing process is again another process is there where you have to macerate the tissue and allow the cells to release into the solution, and that solution or that extract is what you will culture. Okay, then for prolonged survival, a balanced salt solution is supplemented with various formulations of organic compounds or serum. So in case your cell culture, the performing of the culture is going to take a little more time, then this balanced salt solution can be enriched with some more compounds for it to before you go in for the actual culturing process. Then for indefinite growth, also we need this different types of artificial media and specialized functions, as in their specific growth conditions. Okay, so that's the importance of natural and the artificial media. So again, this artificial media, they are grouped into different, that is 4 categories. So 2 are mentioned in this slide. So first one is serum containing media and serum free media. So we've been seeing about the serum throughout from last week. So basically the serum containing media, I told you that fetal bovine serum is the mostly used supplement. Okay, it's a common supplement that's available. Even in our own laboratories, we have fetal bovine serum in our culture lab. Okay, so that is an important supplement for the animal cell culture media. So it's used as a low cost supplement and to provide an optimum cell culture medium. So serum provides carriers or chelators for labile or water insoluble nutrients, hormones and growth factors, protease inhibitors, and binds and neutralizes toxic moieties. So you can see the importance and the functions of this serum in media. So the ones which contain the medias which contain serum has all these properties. Okay, so they have the protease inhibitors in them, and also they neutralize toxic moieties. Okay, so such additional additional benefits are also there if the serum is present in the media. Then if you consider the serum free media, you can see that presence of serum in the media has many drawbacks and lead to serious misinterpretations and immunological studies. The drawback of presence of serum, apart from this additional qualities it also has that is, it can create some serious immunological stress can be created by the presence of serum, because why that is told, because the serum is from another source. Okay, so serum is from a fetal bovine source. So in that way, it might interfere with this the cell's capacity to grow. Okay, so that's why the cells which are compatible, or incompatible for all these things will be grown in a serum free media. Okay, so in the cells are very it requires a very optimum condition, or if cells which have to be isolated requires more better growth medium, that's when the serum free media will be considered. So serum-free media are developed, and the media generally it's formulated to support the culture of a single cell type. So as I mentioned, those cells which are not able to become compatible with the environment, with the different types of nutrients added, or the type of cell which we culture also matters for selecting a serum-free media. And it incorporates defined quantities of growth factors, lipoproteins, and other proteins of interest. And otherwise that is actually provided by the serum. These proteins, I told you, serum is enriched with different types of proteins. So that's why the serum free media should be enriched, that is supplemented with these proteins, lipoproteins, and other important proteins of interest. Okay, so these media can also be referred as defined culture medium. Okay, because that's why, because all the components you add in that serum free media are known to you. Okay, so that's why it's called the defined culture media. So specifically each and every component, you know how much you have added for the cell culture to cells to grow. The other 2 types of categories is chemically defined media. So chemically defined media, mainly that's because you want to have a medium which is contamination free. Okay, and that is, you have to add these pure, that is ultra pure, very pure inorganic and organic ingredients, and may also contain pure nutrient additives. Okay, growth factors and nutrient additives. Only thing is, you should make 100% sure that these are very pure forms. Okay, and these constituents are produced in bacteria or yeast by genetic engineering, with addition of vitamins, cholesterol, specific amino acids, and fatty acids. So they are generated outside and they are added. Only thing, they are contamination free. Yes, the next type of media is a protein-free media. Okay, this is in case of you are culturing a specific cell type again. So protein-free media do not contain any protein and only contain non-protein constituents. So unless and until you need such a type of cell, and you have to grow a cell which is not compatible to the proteins, only then you will have to culture it using the protein-free media. So compared to serum supplemented media, use of protein-free media promotes superior cell growth and protein expression and facilitates a downstream purification of expressed product. So this is in case of when you want to go in for a purification process, or when you go in for a protein expression, you want to, that is why you're making the media protein free, is you are going to express a particular protein of interest. Okay, so that's the idea, that protein of interest should not be interfered by the protein present in your media. Okay, so such when you're going for protein expression studies, so that's why only that specific proteins of interest will be added to the media, whereas the other common proteins which are required for growth will not be there. Okay, that is mainly to prevent the interference. Then formulations like MEM, RPMI 1640 are protein free, and protein supplement is provided when required. Okay, so they're just available commercially without the protein, and we add the supplement whenever we require and whatever type of protein we require. Still, there's any doubt, people? Anybody has any wants any clarification? If, yes, if nobody is having any doubt, I'll continue. So this is the advantages and disadvantages of serum in media.
Ma'am, I had a doubt.
Yeah. Yes, Nimi.
Why exactly is the serum? Why in some mediums that when you have a serum, it's not, very efficient, or I mean, it gives you a misinterpretation. I didn't understand that.
Okay. No, that's because I told you that the serum that is obtained, basically here we use a fetal bovine serum. So there might be interference of factors from the serum with the cells that you're growing. Okay, so there might be like, though it's enhancing the growth, there might be like a immunosuppression kind of a thing, or it'll not bring about the normal growth of the cells. The characteristics of the cells can be changed, as in if you want the cells to grow in with some type of, this is a normal characteristic of the cells will be known, but when you observe them after the growth, you might see that there is change in their characters, as in the size, the shape, there is some disformation or damage done to the cells. So such things can be due to the interference of components in the serum. Okay, so that's why we go in for a use of serum free media. I'll just mention those things in the advantage, disadvantage also. Is that okay, Nimi?
Yes, ma'am.
Okay. So you can see that advantage of using a serum in media is serum contains various growth factors and hormones which stimulate cell growth and functions. Whereas the disadvantage, if you see, it's lack of uniformity in the composition. That's what I did mention, that we are not sure about the actual compositions of these growth factors or the hormones which are present in a serum. Okay, so that's why there might be some kind of disadvantage which is given to the growth of the cells, and lack of uniformity, that's what is mentioned here. Okay, then helps in attachment of cells, that's the advantage. And the disadvantage will be testing needs to be done to maintain the quality of each batch before using. So that's why we are not sure about what kind of changes or modifications the cells are going through due to the presence of the serum. Okay, so that's why before culturing or before subculturing, every time you have to go through a testing. That is, you have to monitor its characters, you have to monitor the size, the growth and the morphology, mainly the morphology, structure, appearance, and also you have to analyze those things, and also counting. That's why before culturing, every culturing, you go in for the cell counting. Okay, that's because to see if your cells have proliferated in a maximum, and given you the maximum proliferation. So such things have to be analyzed before you go in for a culture again. Okay, then acts as a spreading factor. So here serum will act as a spreading factor, and here it may contain some growth inhibiting factors. So you're not aware, maybe along with the enhancement of growth, it might also have a growth inhibiting factors, and acts as a buffering agent which helps in maintaining the pH of the culture media, but it can increase the risk of contamination. So that's why I told you it's from another source. It's from the fetal bovine source. So it might have an interaction with the cells that you're culturing. So that might cause a contamination also. So that's why it is disadvantage for us also. Then the next advantage is functions as a binding protein. Okay. Then the next advantage you see, it minimizes mechanical damages. Sorry. Or damages caused by viscosity. But here, the presence of serum in media might interfere with the purification and isolation of cell culture products. Okay, so such disadvantages are also there. So basically here, advantage is when you always have to choose a serum media or serum free media based on the types of cells that you are culturing. Okay, so you the research that you do based on the cell culture, and as you start practicing, you will also know the quality of each cells, that is how the cells are adapting to the environment, how the cells, what kind of requirements it might need. So based on that, you must always decide your cell culture